gentlemen, good morning. I'd like to extend a personal welcome to you all to London. I am sure the next couple of days are going to be hugely interesting and stimulating. And as a physicist, I'm looking forward to finding out that engineers can actually be quite clever and useful. Dr. J. Craig Venter is a biologist, renowned for his contributions in sequencing the first draft human genome in 2001, the first complete deployed human genome in 2007, and construction of the first synthetic bacterial cell in 2010. He is founder of the J. Craig Venter Institute and founder of the company Synthetic Genomics Incorporated. In 2008, he received the US National Medal of Science and is a member of the US National Academy of Sciences. Ladies and gentlemen, I'd like to invite Craig Venter to the podium to deliver his plenary.
Well, thank you very much, Jim, and it's a pleasure and an unusual honor to be here as a biologist to address an engineering conference. And I think it's even more telling to be introduced by a physicist. That maybe the definition of engineering is changing to something more like evidence-based decision-making versus what the tradition has been. I'm going to talk a little bit about synthetic life and the attempt to control nature. But as the first speaker, I thought it was important to maybe put some of the things in context and show you what's motivating me. And this is UN projections of the population changes. I was born in 1946, and now there's close to 3.5 times as many people on the planet than the year that I was born. That's a massive change during a part of a lifetime. And we're going to add another billion people in the next 11 years. That's almost the equivalent of adding another India or China to the planet in a little over a decade. You can see that India and China represent well over a third of the world's population now. So doubling that number over the next 25 years or so presents a huge challenge in the face of our current capabilities with our existing population, which are not great. I had the privilege of taking part in the National Academy of Engineering program 5 years ago. I'm sad to say the changes in the last 5 years haven't exactly improved the situation. So while there's lots of grand challenges, I think the challenges keep getting grander. India is going to overtake China as the world's largest population by 2030 and is growing substantially beyond that. I have a simple equation that's what is governing some of our thinking, and that's shown here, that fuel, water, and food are essentially equivalent because we need all three, and one can lead to another. But when we look at the energy side, for example, we're using over 75 million barrels of oil a day. Just that number alone presents a fantastic challenge when we think of trying to replace that with some other source. Coal's even worse, 4.5 billion tons a year that's burnt. And of course, the largest population countries use the most. Burning all that oil, burning out all the coal has led to a pretty steady, if not increasing, CO2 concentration in the atmosphere. This is the famous Keeling curve showing those changes. We don't know for sure what the impact of this is going to be, but it's kind of like playing Russian roulette and hoping for the best. We've sort of saturated the carbon sinks now the ocean being the largest carbon sink, but that has now been saturated, and therefore the atmosphere is the only place for this excess carbon to go. I like to think of carbon, particularly in the form of CO2, as the potential greatest natural resource for the future, and I'll get into that in a minute. But when we look at the scale of the problem with carbon dioxide emissions, the northern hemisphere, particularly with the United States, with China and Europe, contribute well over half of the problem. And in fact, just the change in 4 years in China contributes to most of the increase that's been seen in the last 4 or 5 years. So an increasing industrialization with increasing populations is not improving the situation. And this just gives an example with one country, and I'm not picking on China because this is happening on a global basis, of dramatic shifts in CO2 is just one measure of how we're changing the environment. Water, as some people have mentioned, is one of our greatest challenges. Typical use averages around 5,000 liters per day to produce food. In the Mideast, it's more like 5,500 liters. And most of the freshwater use has gone into agriculture. But now with water for extracting natural gas, with fracking, and with oil, we're increasing using a greater portion of water just to produce energy. So the water is driving the energy side. To produce animals for food, just in the U.S., it's about 10 billion tons of fertilizer, mostly driven by oil. But as you enjoy your steak this evening, keep in mind that it takes about 10 kilograms of feed, 1,500 liters of water to produce 1 kilogram of beef. It's clearly not sustainable, and it's not expandable for the billions that we're adding. But currently we produce about 280 million tons of beef by converting those animals on the left into the meat counter on the right. I'm afraid it's becoming a cliche word without the meaning behind it, and that's disruptive change. I've heard the term about 10 times in the last 24 hours, but I don't see much evidence for disruptive change, and I find that extremely discouraging. One thing that could be disruptive is the advances in synthetic life, and so I'd like to walk you through those changes because they can impact every part of humanity. And maybe this is where engineering fits into the bigger picture, but I view science, and including within science as engineering, is something now we're 100% dependent on for our future. Much of the science today is needed to overcome the science and engineering problems from the past. 100 years ago, it seemed like a great idea to use oil out of the ground instead of killing whales to produce oil to light lamps. I don't think anybody anticipated the problem of burning millions of barrels a day and increasing and changing the entire climate of our planet. At my institute, the not-for-profit institute in California, we're actually using some real engineering, traditional engineering, to try and create the world's first carbon-neutral research building with water-cooled computers and all kinds of novel design. This is a computer-controlled building, and it will be coming online in October of this year. And within this institute, we're actually engineering cells at the smallest level. but it has been a goal of humanity since the earliest stages of civilization to get control over nature. This happened with domestication of animals. That happened with agriculture. And we haven't changed much in what we do, just the scale of what we do since the earliest parts of civilization. We now have a chance to change that with the realization, again, in the last 70 years, that DNA is the software of life. Prior to 1944, the scientific community was convinced that proteins were the genetic material. So everything we know about modern biology has happened also in my lifetime. In 1977, Sanger's team read the first DNA of a virus, and ever since that time, as we take this 4-letter genetic code and read that code and add it to computers, we're doing what I call digitizing biology. We're converting the A, C, G's, and T's into the ones and zeros of the computer. And that's been happening at a grander and grander scale. My team sequenced the first genome of a living cell in 1995, and 5 years later scaled that up to the first draft of the human genome. And the databases have just been working at a multiple exponential phase and adding more and more data. The majority of gene sequences have actually come from a simple expedition that we did called the Sorcerer Expedition, where we just sampled seawater every 200 miles around the globe, sequencing the organisms that we were able to filter out, dealing with it as digital data and recompiling these organisms in the computer. We've now discovered over 80 million genes in untold new organisms through this process. So we have large databases, and they give us a very different view of the world. What people thought were single organisms in the ocean, in our lakes, turned out to be tens of thousands of related species, but not even very closely related. We're related to all mammals with about a 10% change in our genome. These organisms have up to a 50% change in their genetic code, driven largely by oxygen and UV light. And as the data accumulates, what people thought were single cells and single organisms turn out to be whole huge new taxa that a decade ago we were completely ignorant of. So we're still characterizing life and biology on this planet. Probably most of it still remains to be discovered. But these discoveries give us new tools to deal with the future. And we set a new challenge, and the challenge was to start now with the digital code in the computer, the ones and zeros, and see if we could recapitulate life from those ones and zeros, actually proving, in fact, that DNA is the software of life. I'm going to walk you through those experiments that led to this cover of Science in July of 2010. We started with simple questions. How many genes are essential for life? Can we get to a minimal cellular machinery? And over trying multiple experiments, we got to the point that by building a synthetic chromosome would be the only way to try and answer these questions. But nobody had ever made a synthetic chromosome before. We weren't sure if the chemistry would allow it, or even if we made it, could we find a way to boot it up? So we started, after we finished sequencing the human genome, and a team with the Nobel laureate Ham Smith and Clyde Hutchinson and myself, trying to make a small bacteriophage virus that kills bacteria, and starting with its digital code in the computer, 4 bottles of chemicals, and finding ways to correct the errors in the chemical synthesis. We made this small piece of DNA replicating the genome of Phi-X174 and injected it into E. coli. And what we found is this synthetic piece of DNA was read as normal DNA. The proteins were produced, they self-assembled and formed the virus. And the virus replicated and killed the cells that made it, which is how we detect it with these clear plaques on a on a bacterial lawn. So I call this a case where the software is building its own hardware. All we did was put in a piece of DNA software, and that turned into a viral particle that had unique biological activity. Our goal was to make not a virus, but an entire bacterial chromosome. But we thought if we could make a virus, we could replicate that process over and over and find a way to put the pieces together. To make a long story short, over several years, we developed technology for assembling large pieces of DNA. And we could take 4 synthetic quarter molecules, just put these into brewer's yeast, and yeast links all these pieces together. And that gave us our first synthetic cell. Young scientist Dan Gibson took the procedures that our older team had developed and converted into a single-step reaction. And this is now known around the globe as the Gibson assembly. And what this allows us to do is now automate this process. So we can go from just the digital code in the computer to the biological code in a robotic fashion. So we could make large pieces of DNA. Can we boot them up as you can new software code? And this study that we published in 2007 is perhaps one of the most important in this direction. By simply changing the DNA in this cell, we can convert one species into another. So we isolated a chromosome from one cell, and we inserted it into another. And so now we have a species with a phenotype, one cell, and it has 2 bits of genetic instructions. But this was designed so as the new DNA was read, it produced enzymes, restriction enzymes, that recognize the parent chromosome in the cell as foreign material and chewed it up. So now we have the body and the phenotype of one species and the genetic instructions of another. In a short time, these cells were converted into what the new genetic instructions told them to do, and every trace of the pre-existing species was gone. So life is a DNA software system. If you change the software, you change the species. So we wanted to do this now with a synthetic chromosome. With new technologies, we built this 1.1 million base pair genome and went to do the transplantations. It didn't work. Ultimately, we found just one letter being wrong out of 1.1 million made the difference between life and no life. We corrected that error, and in 2010, we got the first synthetic cell. Built into this genome was a URL. As the first species with a parent as a computer, it should have its own web address. And this was all reported, as I said, in Science. So where are we today with synthetic genomics? I view these 80 million genes discovered as design components, much in the same way the electronics industry had resistors and capacitors, then eventually transistors and integrated circuits, only we have tens of millions of these design components. We actually have computer software for designing new biological software to get biology to do what we want it to do. So we're designing new cells to use CO2 as its carbon source, use light as its energy source, to take us in new directions for food, chemicals, plastics, etc. So future foods and medicines will utilize not only information from your genome and microbiome, will use information from stem cells made from your skin cells, but can avoid using animals as a source of feed. We can replicate everything nutritious in a much more efficient fashion. If you look at the production of plants compared to what we can do with microalgae, even the best producing plant of oil palm is small on scale to what we can get with algae cells. The trouble is existing algae cells don't have the ability on their own to produce what we need on scale for food and fuel. So we're taking a synthetic chromosome approach to add new attributes to these algae cells, and we've had some success. For the first time ever, we've been able to alter photo efficiency increasing it threefold. So we get 3 times as much energy per photon has been impossible from natural evolution. We can scale this up in greenhouses and take them to larger production facilities. You've been hearing the news about antibiotic resistance. This is now a bigger problem globally than most people realize. More people die from resistant bacteria in the U.S. than from car accidents. And it's finally reaching here, making public news. There's new approaches, ones that were tried a long time ago in the former Soviet Union until penicillin came along. And that's using things like Phi-X174, the viruses that attack specific bacteria. Only we're now making these synthetically, designing the genome to have very specific bacterial repertoire. The first genome-based vaccine just got approved here in Europe earlier this year, and that's the one we made with Reno Rapalli at Novartis against type B meningitis. Meningitis is one of those diseases, by the time you diagnose it, the young people die. You can prevent it with vaccination. And we've now turned that to flu vaccines, because as we saw with the 1918 pandemic, about 3% of the world's population were killed by a single virus. The chances of this happening today are calculated to be even greater. Currently, we use about 900 million eggs just to produce the annual vaccine doses for the U.S. So new technology is coming from us sequencing the genome of emerging viruses, tracking this in databases, and now we have a process where we can actually synthesize using synthetic DNA a new viral candidate for a vaccine in less than 12 hours. In fact, it starts with digital information. BARDA and the U.S. government sends us a test pandemic sequence. We have 12 hours to make that and get it to the new Novartis facility to start manufacturing. So we're pandemic ready with this process, and we call this a digital biological converter. We can take digital information off the internet and convert it back into biology. This is the plant now that just got approved in North Carolina using cells instead of chicken eggs to produce a vaccine. But this can be replicated around the world. We can send a new flu vaccine around the world in less than a second digitally. And with a digital biological converter, you can convert that back into an actual vaccine. So the idea will be to build centers around the world, but ultimately getting even smaller. Think about this in terms of getting samples to and from Mars, from Earth. This is a huge engineering challenge that people talk about spending hundreds of billions of dollars to bring samples back from Mars. A much simpler thing would be to send a DNA sequencer there, sequence whatever organisms, and send them back as a digital magnetic wave in as little as 4.3 minutes, and rebuild the Martians in a lab here instead of having them arrive and crash down into the ocean. A lot of people are afraid of alien life, although we exchange about 100 kilograms of material between Earth and Mars, so I think we're already invaded. Everybody's aware of 3D printers. We're changing the whole manufacturing process, now including biology, to sending digital information and converting it on-site into materials or into biology. We can now print skin, and other tissues are underway. The future will be having a small box attached to your home computer, where if you have diabetes, you can download insulin from the internet. If there's a new flu pandemic, instead of waiting as what happened with H1N1, where the vaccine only became available after the peak of the pandemic, you can download it in your own home, and we can eliminate future pandemics. So we call this biological teleportation, sending biology at the speed of light. It changes the ability for any nation, as long as they have access to digital information, to do their own manufacturing. The key to the future will be the cost of raw materials and intellectual property. Everything else will be totally doable in a remote fashion. Just to close, we've undergone constant ethical scrutiny of this, asking for the first reports in 1999. The Sloan Foundation has funded my institute along with MIT and others, including input from the EU on biosecurity governance. And when we made our announcement in 2010, President Obama asked his new ethics committee to take this on as their first challenge, and this report is available from government sites. These are huge challenges, but bringing it back to engineering and to your own academy, this is a report on synthetic biology in 2009 saying this could be the technology that actually revolutionizes much of what we do on the planet. I think that's even more true today, and I hope that biology will be a key part of your future. Thank you very much.
Thank you very much, Craig. Well, I invite you to join me here at the table. We'll open up a Q&A session. Now, I know the program says we're due to break at 10:10, but since Craig was late starting, we want to have enough of an opportunity for some questions. So I propose we eat 10 minutes into the break. I'm sure 20 minutes is still enough for a comfort break and a coffee and a quick chat. The format is that you should raise your hand if you have a question. Please wait until a roving mic arrives, and then if you could give your name and affiliation before you ask the question. I guess it's my prerogative as chair of the session to maybe start up with one question. We could have the house lights up as well, so that yes, so we can see where the raised hands actually are. Okay, thank you. Can I just ask you a quick question, Craig? You were on the original committee put together for the NAE's Grand Global Challenges, and writing the report. Do you see that the agenda has moved on in the few years since then?
I think everything that was reported back then is even more true today. I think what's discouraging is the lack of progress, perhaps in the last 5 years. And progress at least has to keep up with the pace of growth of the population and the changes that we're seeing. So just between now and the next meeting, there's going to be a few 100,000 people added to the population, right? The problem's getting bigger much faster than we're doing anything about it.
Okay, so let's have, yes, okay, so we have one question over here, and then another one towards the back there. Yeah. Sorry, oh, sorry, no, you've just behind you, second row. Sorry, could you stand up so that people...
And the mic,
sorry,
the mic's on.
My question is, proposing synthetic life. Seeing the reaction against genetically modified food, do you think population will accept it?
I think it's an important question, and I usually answer that facetiously of we're trying to design DNA-free food for Europeans, but I think the rest of the world is more than ready for it, yes.
Thank you. Okay, so there's a question. Yes.
Hi, I'm Jack Langlad from Penn State. With transmitting these DNA recipes, presumably over the internet, is there going to be a problem with security of the information, right? Putting in rogue code, for example, in someone's medicine that's getting transmitted. How would we be able to address something like that?
Yeah. No, it's a very important question. It means we need to change our definitions of antiviral software.
Okay, so gentleman there, and then in the middle here with the raised hand, and then I want to see if there's something on this side.
Yes. My name is Hari Pujar from Merck & Company in the U.S. Craig, could you hazard a guess as to when 3D printing of vaccines and insulin, as you mentioned, would actually turn to reality?
Well, it's a reality today in our laboratories. You can see why the FDA might have a little bit of difficulty of, bypassing the huge infrastructure associated with drug approval and distribution. But we have to bypass it. As I said, with the H1N1 vaccine, as you know, the vaccine only came out after the pandemic had peaked. And if it had been a real global pandemic that affected just a few limited number of previously non-immunized people, it could have been a global disaster. So we need to change it very quickly, maybe just for pandemic readiness now. I think the problems with all these technologies are going to be social and regulatory rather than the technology, biological, and engineering challenges. We could set up a receiver now in Africa to send new antimicrobials there. Mars will be colonized within 2 decades. The fastest way to get information and materials to and from Mars is going to be electromagnetic waves, not spaceships. So I think it's a question of how fast these things can be implemented and what's the regulatory structure that builds in around it, including, like the previous question, the safety aspects and the antiviral software.
Can I just say before we move on, for those watching this in the UK and India, I have an iPad here, so I'm going to be sent questions from the other sites. So I do encourage you to send your questions in. I'll try and incorporate them. So we had a question there. Yes.
So, hello, my name is Hugh. I'm from the University of Southampton. So I have thank you, Dr. Craig, for your very inspiring talk. I just have, I think synthetic biology is very significant. So I think it's analogous as making robots. It could help the mankind, but it can demolish the whole mankind. So I wonder if anything that if any measure you're taking to protect synthetic biology from killing the whole mankind. For example, making a virus can be made and it kill the whole mankind on the world. So if any measure to make it safe and control the way.
So I think your question illustrates one of the biggest problems in our society, that we get our science education from science fiction movies, not from anything based on reality. The worst pandemics of very potent viruses, the worst they've killed is a few percentage of the population. Trying to design or accidentally making a virus that will wipe out humanity is a silly notion at best, but there's constant safeguards also built into things in terms of we remove the genes associated with any infectivity in the organisms we're working on. They're designed not to live outside of a laboratory or a manufacturing facility, but everybody has to follow the same guidelines.
Okay, well, now the hands are coming up. So there's a gentleman in the third row who is, and then on the side there.
Subhash Mahajan, University of California, Davis. The increase in population is a social challenge. How synthetic biology will help in resolving or solving that social challenge?
Well, the social challenge, as evidence has shown, as countries and populations increase their social economic standards, reproductive rates fall quite dramatically. So I think one of the main social challenges that's been mentioned already by others is improving the economic and health environment of the developing world instead of having it just be part of the first world at the expense of the rest of the world. I think populations, people want to get their own populations under control when they have the opportunity to do that. When death rates fall from children, when they're not needed for manufacturing and farming. So it is a social problem, but it's a social economic problem that modern tools of science and engineering can help change that inequity.
question over there, and then...
Okay, I'm Jimmy Zhang from Chinese Academy of Engineering. So you made the first step to use a synthetic approach, making the bacteria. So considering the human genome is so complicated, so do you think it's possible, or how soon you can reach that one, use this approach in the like human engineering, tissue engineering?
Yeah. So people always want to make that kind of leap from engineering bacteria to engineering humans. You know, I live in California and Washington, D.C. We have enough synthetic humans already. You know, so I don't think we necessarily want to engineer more humans. Or even, you know, nobody knows necessarily how to make better ones. So, but we live on a microbial planet. We have more microbes associated with our bodies that affect our health than we have human genes. You know, manufacturing can be totally changed by simply engineering microbes. Our oxygen that we breathe comes from these microbes in the ocean. So don't discount the importance of microbes to your life and to your future. Engineering of DNA is engineering of DNA, but human experimentation is now something that's been eliminated largely from science, other than under well-controlled clinical trials for the benefit of humanity.
Yes.
MIT. You talked about sort of the slow advancement over the last 5 years, and I'm wondering, in your thoughts, what are the barriers, and what kind of structure would you envision? You mentioned sort of advancing engineering challenges, but also the social regulatory. So what kind of global collaborative structure would you envision that would move that advancement much farther, and what are those barriers that you faced?
I mean, it's a great question, and just pointing out problems versus solutions is a challenge. One thing that's allowed my small institution to be very successful is it started de novo, and there's no departments or walls or barriers. There are multidisciplinary teams. So our sequencing of the human genome in 9 months was because we had engineers, electrical engineers, computer scientists, software engineers, biologists, molecular biologists, chemists, all working together on the same problem for a common goal. The U.S. government wouldn't fund it because it was too risky. So instead of doing it for $100 million, they spent globally, including here in the UK, $5 billion to accomplish the same thing over a much longer period. We need to change the way we fund basic science in the U.S. I think at least 90% of it is largely wasted because we won't fund new unknown things. We only fund what's known. So I think going to multidisciplinary teams was something like grand challenges. That's why I like the grand challenge approach that was the Academy of Engineering came up with. Because what I found with the Human Genome Project and others is most people feel more satisfied working and contributing their knowledge to a project much larger than themselves than the typical academic of working in your own isolated lab on your narrow little piece of the world. And I think not only would it improve the quality and the pace of change, we would have probably 10 to 100 times as much for our science funding as we get today. I'd at least like to see that experiment tried.
Okay, so 2 questions from the back, and then 2 from the front, and then that'll be it.
Dale Carter, University of Strathclyde. Do you see using synthetic biology to solve things such as Alzheimer's and cancer? Hi. Yeah, do you see the work that you're doing in the future is going to be able to solve such things as Alzheimer's and cancer in the future?
I'm not sure synthetic biology per se will be. I think having the human genome sequence and using that to understand what parts of those are genetically based, which part are environmentally based, will have a major role. We're still at a very primitive stage understanding our own human circuitry. Having the human genome sequence as a grand challenge provided a new basis of how people are approaching that. There's lots of money and lots of time going into these issues because in developed countries, diseases like you mentioned, Alzheimer's, dementia, is going to be one of the biggest expenses for society. If you don't die from heart disease or cancer, your chances of getting dementia are very high.
Okay, so we don't have much time, but I'm going to squeeze in 4 more questions. So yes, lady at the back then.
I'm Debbie Chatra at Olin College in Massachusetts. Dr. Venter, you said that in the future the issues are going to be raw materials and intellectual property. I wonder, given what we know about sort of current digital copyright and also about pharmaceutical patents, if you could speak more about that second point.
Well, that's a huge landmine, but I think, much in the way that the music and television and movie industry has had to adapt, I think the pharmaceutical and other industries will as well. When you buy drugs from your pharmacist, you're paying all these fees. It's no different than how you download things from the internet. So I think it will just change how it's paid for and how it's regulated. I think intellectual property will become more important for the future than it is today.
Yes.
Hi, my name is Sujata Bhatia, and I'm from Harvard University. And my question for Dr. Venter is, do you think that synthetic biology could be used not only to design new life and design new raw materials, but also to have a better understanding of existing raw materials on the Earth and how we can better leverage existing materials to sort of create a better taxonomy of materials for sustainable development and health, and sort of better understand the resources we have at our disposal?
No, it's a great question. I think what it can do is lead to a better understanding of the biology of our planet. We've discovered so many genes just from organisms in the oceans. There's not enough scientists on the Earth right now in 100 years with traditional science to work out what they do and what their role in life is. But with synthetic biology and the robots that we're building, we're trying to build a self-learning robot that can make constructs and test them and learn 10,000 times faster than a scientist can. We need to do that because that's the problem with the future of design. We can't do truly intelligent design of life without understanding what all the components do. And that's true even for the most basic cell we're trying to build in the computer right now. Whether it can help with raw materials, I think it will indirectly with that.
Yes.
I'm Amos Avedon from Bechtel. Dr. Venter, I have a question for you on when you look at the basis of a lot of the grand challenges today or in the past, they seem to me many times to be based on this concept of scarcity, going back to Malthusian projections and the Club of Rome, and all, as if we are running out of a lot of natural resources. And you still hear a lot of that in Europe, which is probably understandable when you look at the outcome of the fiscal crisis, mismanagement, and things like that. But what I get from your talk and from a lot of others is not the scarcity of resources, but it's the proper management of resources, new technologies, R&D, sustainable development, which are what you're focused on. Would you comment on that, please?
Well, there are some resources that we are depleting. When you fish out the oceans, which we've done to the extent of about 99% now, it's hard just to replenish that resource and have that source of food. So it depends on what you define as a natural resource. My good friend Peter DeMontes has formed an asteroid mining company because the ability to bring in raw materials for manufacturing from nearby asteroids sort of eliminates some of the scarcity issues. But if we keep doing what we're doing with our environmental issues, fishing out the oceans, changing our own climate, what most people consider the raw materials are going to be the least of our problems. So there's lots of ways to replenish them from this planet or from nearby resources. And I think asteroid mining and mining on other planets is not science fiction. I think we'll see that in the immediate future. It's still a question of, with our ever-expanding population, how to manage these fundamental resources. How do you feed people? How do you provide the medicine? How do you prevent pandemics in overcoming all the problems we have with just producing the basics? Clean water is going to be one of the biggest problems for the future. So that's a tremendous natural resource issue.
Okay, thank you very much. I think I'll bring this session to a close. Please join me in thanking Dr. Craig Venter for a fascinating beginning to this event. Brilliant. Thank you.